Okay, this is the overall nervous system review. So I want to point out some important terms that we have here, and I'm not going to read this word for word or anything, but not all organisms have a brain. Sometimes there's a cluster of neurons called a ganglia. We in the human body have ganglia, and we have, of course, a brain. And some don't have either one of those. There's no clusters of those. We just have cells that reach threshold and can actually respond to a stimuli. And depending on how they are organized will be how the one signal passes to the other. And pretty much the three major parts of the nervous system are sensory, and then an interpretive part, and then a motor area. So here is a squid, which is very interesting. Squids are associated with octopus. They're in the these are the cephalopod, actually. The cephalopods, they belong to the mollusk families. What I mean by that is, so when I say cephalopods, so I'm talking about like clams, that area. They are not echinoderms. They're in like the mollusk family, like with snails. And I'm trying to remember what else. Octopus, squid, they're all in that same area. So you see here, the squid does have a brain, and here's its eyes, and here's a bunch of its nerves that it has in the tentacle, the arms, and this is the mantle of the body, but you don't have to remember that. So here's neuron or nerves, and then here is like the ganglia, so groups where they associate. Kind of interesting. So it's eyes as far as sensory, for light interpretation or sensory to light. So you can kind of think about sensory neurons as being the center to the football team that gets the football, and then they pass it on to the quarterback, which would be your interneuron, that's actually going to determine where to take that information to, and the motor neuron would be either the running back or the wide receiver that would get the ball and actually do something with it. This is very primitive, but this is so explains like a reflex arc in the body where you get the senses, okay, my hand is on a hot stove, I feel pain. This goes to the spinal cord where the interneurons then shoot the message back out to your arms to get you to contract and pull your hand off from that hot stove. Your central nervous system is, of course, your brain and spinal cord, and your peripheral nervous system is everything outside of that. And so here's a great example. In the interesting part, not everything goes right to the brain to be interpreted. Sometimes it goes to the gray matter, which is in your spinal cord, to go back out. That's what a reflex arc is. It happens so fast that it can go out. It doesn't need to go to the brain. There are also, I wanted to point out, there are also parts in the brain that serve as interneurons that actually shoot the message out to different parts of the brain depending on where it needs to go. We already pointed out the cell body and the dendrites that actually receive the signal in the axon, which can be huge, and pass on that signal. So it goes from dendrites to axon. And here we go. Here's the stimulus goes in here and then passes along. So from the looks of this is not myelinated. If it had myelin, it would have the fat that's located around here. And you don't have to remember the actual name of the cells that wrap their cells around here, but this is very long. Function is directly related to the shape, so there's a reason why it's so long. There are also different shapes of neurons besides just this one. This in between, of course, the gap is called the synapse. They don't touch one another, but they're really doggone close to transfer on those neurotransmitters, those chemicals. And there can be a bunch of different particles. So I'm not really too concerned about you knowing presynaptic or postsynaptic between the two. There are types of cells. We call these glia cells. We call these the maids and butlers. Neurons are so special that they need maids and butlers to take care of them. And there are 4 major types of glial cells that actually like take care of those neurons, eat up dead stuff, bring blood vessels to them, form coating, forming membranes. So, but you don't have to know those. Nor do you have to look at the shape of these guys. Okay, that's not necessary. Okay, so this is what I was talking about in the back of my room with the big pieces of paper was looking at resting potential versus action potential. And on the outside of a mammalian neuron, you have a concentration of sodium on the outside and potassium on the inside. And it causes a different gradient. It's polarized. The outside tends to be positive and the inside tends to be negative because of some other ions that are also located. Sodium can go in and out, but it takes ATP in order to do that. So it does, the sodium potassium pump is active transport, and it does take energy to do that. But the potassium tends to go in and out a little bit easier. Of course, they go through ion channels. Again, fewer open sodiums and the potassium. So the sodiums tend to stay outside, and you get that negative charge on the inside. This is called resting potential. I've lost my... There we go.
A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. The cytoplasm just inside the membrane is negative in charge, and the fluid just outside the membrane is positive. The membrane is said to be polarized. Like a flashlight battery, the difference in electrical charge carries potential energy, which can be measured as a voltage. The oscilloscope shows this charge difference, called the resting potential, in millivolts. A neuron has a resting potential of about minus 70 millivolts. What causes the resting potential?
The plasma membrane of the neuron keeps large negatively charged molecules such as proteins inside the cell. Pumps and channels in the membrane regulate the flow of positively charged sodium and potassium ions. The neuron maintains a higher concentration of sodium ions outside the cell than inside, and a higher concentration of potassium ions inside the cell than outside. In a resting neuron, most sodium channels are closed, but many potassium channels are open. As a result, very little sodium can diffuse into the cell, but potassium ions can flow freely out of the cell, adding to the positive charge outside. The sodium-potassium pump actively transports more sodium ions out of the cell than potassium ions into the cell. This contributes to the buildup of positive charge outside the cell, while keeping the concentration of sodium low and the concentration of potassium high inside the cell. The diffusion of positive potassium ions out of the cell and the large, negatively charged molecules trapped inside the cell cause the charge difference that constitutes the resting potential.
A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. The cytoplasm just inside the membrane is negative in charge, and the fluid just outside the membrane is positive. The membrane is said to be polarized. Like a flashlight battery, the difference in electrical charge carries potential energy, which can be measured as a voltage. The oscilloscope shows this charge difference, called the resting potential, in millivolts. A neuron has a resting potential of about minus 70 millivolts. What causes the resting potential?
The plasma membrane of the neuron keeps large negatively charged molecules such as proteins inside the cell. Pumps and channels in the membrane regulate the flow of positively charged sodium and potassium ions. The neuron maintains a higher concentration of sodium ions.
Okay, sorry about that. Okay, sorry if that repeated on you. So here's a picture of most of your sodium out here and most of your potassium in here, and your different pumps. So this again is resting potential. And so the big question is, so what does that mean as far as resting? Well, let's look at some of your numbers here of the difference in charges. So outer chamber versus inner chamber. This is membrane selectively permeable to potassium, and the membrane selectively permeable to sodium, and how that would change. Kind of interesting when looking at sodium chloride versus potassium. I would not have you do any of these as far as voltage on the test, so we'll go past that. Or we're not going to look at the equilibrium potential. Okay. All right, so action potential, what happens is when the membrane actually, when the neuron reaches threshold, the gated ions will change and actually open up. And so what happens, hyperpolarization and depolarization, is this difference in charges. So depolarization, without getting into all of these graphs here, what actually happens, let me give you action potential. I was hoping they would show me the actual difference between the two. Let me go to this one. Okay. What's going to happen here is that when you reach threshold and the gates open, the sodium is going to rush in, and it wasn't able to rush in before. And so what you will have is a difference in charges. There's no difference anymore. It's positive on the outside and positive on the inside. That's called depolarization. And that will travel down in the open areas. So say, for example, we have myelin over an insulation over the axon in that location. What will happen is that it will have this opening of these gated channels in between where there's no covering. If this is an axon that has absolutely no covering, that it will do the down the entire membrane from the dendrite all the way down to the axon. The minute that happens, the membrane is going to right itself. So all of that sodium rushes in. It's going to let the potassium rush out to repolarize itself. And that will travel right after all of the action potential. So right after you do action potential, then you'll have the resting, go back to resting again, all the way until you get to the end of the axon.
We have looked at the resting potential in a neuron that is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus may change the permeability of the cell membrane to sodium or potassium ions. This alters the distribution of charge in the cell body. And if the change is great enough, it triggers a signal, called an action potential, that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close in response to voltage changes across the membrane. When no action potentials are being transmitted, most of these channels are closed. An action potential begins when voltage-gated sodium channels open and sodium ions rush into the cell. The plasma membrane becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close, while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The plasma membrane returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. The efflux of potassium ions during the falling phase of the action potential restores the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting distribution of sodium and potassium ions.
Okay, so that's going to go, again, until it gets to the synapse. Here's an example, so action potential, and you can see how it goes along the axon. In this case, there is no myelin over top, no insulation. So it's literally going to go down the entire area. I'm sure you've heard of white matter versus gray matter. White matter is those that do have insulation, kind of like the insulation on the coating of the outside of a cord. That will speed up the transmission of this because it's going to jump from open area to open area, and the open areas are called the nodes of Ranvier. That will speed it up. Usually the myelin is found in neurons in the PNS because we want to go from the fingertips all the way into the spinal cord and up to the brain as fast as we can. But there are certain areas of both the spinal cord and of the brain that are gray matter that don't need, and a lot of those are the interneuron areas that are very short, that are just connecting to some other motor neuron, because you literally could have a neuron that is super, super long. So it might take a little bit longer. And when I say longer, we're still in like milliseconds to be able to carry a control. So here we go, the action potential, and then right behind it, we're going to write ourselves. Now, something you might want to think about, well, wonder if you have a conduction going and then you have another one coming right behind it. And that's entirely possible because it's not necessarily, like say you have a really strong stimulus, it's not like you're going to get a different outcome. It's not going to have you might have more neurons stimulated, but you're not going to get a difference in what's going on here. We actually call that the all or none response. Your muscle contractions work the same way as your neurons. Once you reach threshold, okay, that's it. So for example, you have so much patience until you like can't take it anymore, and then when you snap, well, that's the threshold. You either react or don't react. You either send the message or you don't. Okay, so here's where we were talking about the myelin sheath. I'm not going to have you remember the difference between oligodendrocytes in the central nervous system versus Schwann cells. Schwann cells are more prevalent because we're out in the PNS, and here's how we remember a Schwann cell. The Schwann's man brings all kinds of yummy ice cream, which puts layers of fat on everyone, and so I kind of think of the Schwann cells as putting insulation over top of the axons. That's what I always tell. So here's the example. So theirs would be the nodes of Ranvier where you actually have, so the swapping of potassium and sodium happens here and here and here, and so it jumps. So it's much, much faster. Okay, and we're going to get all the way down to the end. So we want to talk about the synapse and the synaptic cleft. So here we go. I don't want to loop. I do want to, there we go, play.
Neurons communicate across junctions called synapses. Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse?
The synaptic cleft between the synaptic terminal of ascending. There is a narrow gap, the synaptic cleft, between the synaptic terminal of ascending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
Okay, here's another great example. So right at the end, everybody thinks calcium is just for bone growth, and that's absolutely not true. Calcium is used for this part where we actually get the message of the neurotransmitters out in the presence of calcium, but calcium is also huge in muscle contraction. So calcium rushes in and causes these synaptic neurotransmitters to bind to the outside and then dump their neurotransmitters, which then are these ligand-gated ion channels. We've talked about this before, which allow, so here you go, here's your neurotransmitter that changes the shape and allows for the rushing in of sodium. So kind of like that helper that changes the shape of the protein, which is pretty cool. And that, ligand-gated, keep in mind that this would also be different depending on the neurotransmitter. There are neurotransmitters that both excite or inhibit, for example. Excitatory postsynaptic potentials. So there are neurotransmitters. So, for example, acetylcholine is a big one that will excite. Caffeine is a drug. It's not a neurotransmitter, but caffeine is a drug that allows for those, I guess, to lower the threshold of neurons. So it makes you hyper, makes you, but ironically, kids that have attention deficit disorder a lot of times take Ritalin, which is a stimulant, but it works backwards. It's kind of interesting. They've looked at dopamine, they've looked at serotonin. Those are all types of neurotransmitters that can inhibit or can excite. So both would work. If I want to come back, see if they have let me come back to here. So, for example, if you want to stop the nerve conduction, at this point, what needs to happen, if there are neurotransmitters in the gap, you either need to reuptake them, which you can. That would be like phagocytosis, taking them back in, or you need to break them down out in the synapse, which you can do by an enzyme that is, for acetylcholine, we call it acetylcholinesterase, but you could have different ones depending on the type of neurotransmitter. You'd have to break those down to stop the nerve conduction. You can get to the point, for example, in an epileptic seizure, where you actually exhaust all the neurotransmitters out of an area and it causes the seizure. So it was kind of interesting. I read that. Okay. Isn't that a great picture? So the synaptic terminals, the presynaptic neurons, I thought that was interesting. And we don't have to know about summation or anything like that. So I think this might be, here's acetylcholine. Interesting. So vertebrates have two major classes of acetylcholine receptors. Acetylcholine can be found in vertebrates and invertebrates, which I think is interesting. You might have heard of some of these. You don't have to remember these, but you might have heard of some of these amino acid transmitters, glutamate and gamma aminobutyric acid, so GABA. You might have heard of some of those. How about epinephrine, norepinephrine, dopamine, serotonin? Epinephrine and norepinephrine, that's adrenaline and noradrenaline. Active in the CNS and the PNS. I'm sure you've heard of endorphins. These are a natural painkiller, very similar to opiates. So you have a natural painkiller that's actually an inhibitor, so it stops the pain. But then, of course, sometimes we need more. So morphine, what was I thinking? Not Dramamine, Demerol, Vicodin, all of those would be types of drugs that would have inhibitors. How about anesthesia? Anesthesia is a great example of being a pain inhibitor, so stopping that pain impulse. So this is kind of interesting, the type of, so morphine is an opiate, and all of these, we know these opiates are definitely addictive for people. So that's all I wanted. Nitric acid and carbon monoxide are local regulators in the PNS. I'm not really interested in that. We're going to stop there. So as far as brain material that I would kind of be interested, this was kind of interesting. If you have time, we were looking at this in physiology. This was a brain tumor. The music to this is pretty wild, but here's the brain tumor, and they actually like cut the scalp and they show it's actually 4 different parts. And the parts of the brain I was going to go to that for a second, but the parts of the brain you have to know is the cerebrum, the cerebellum. Let me see if I can go back. The cerebrum, the cerebellum. I want to do a picture of, hang on. Cerebrum, cerebellum, and the medulla, if you're not aware of those. Not all creatures have the same. There we go. I want to get a picture of the brain. Here we go. Here's a great picture. And I'm going to... Okay, so this is all of your cerebrum. Actually, all of this is the cerebrum. These different areas that they're pointing out here would be some of the areas that of the cerebral cortex that actually different areas. For example, vision's in the back and hearing is on the side, and the frontal lobe. The Broca's areas for motor and speech. Smell is towards the inside. You don't have to remember those areas. I'm not interested in that. This is all the cerebrum. It is formed this way, and it's all have all these convolutions to increase surface area. You have the cerebellum, which is in this area, and then here's your medulla, pons area. This is your area right down here that's super important. This is all of your involuntary control, your heartbeat, your respiratory, your blood pressure, all of that. This is what would get squeezed down through the bottom of your skull. If you had meningitis and it started swelling, that would be that area. From right here, when your nerve impulses, then they shoot off to the different areas. Deep within the brain, of course, is your glandular stuff, like your pituitary gland, and your hypothalamus is like, if I did a cross-section, I don't know if they have a cross-section. I was going to see. Cross-section through the center of it. That's where that area would be. Let me see if I can do a cross-section, like right here. In this area right here is where you have, this is your pituitary gland down here, and your hypothalamus and thalamus is hypothalamus here, thalamus here. This is all of like your glandular material. So here's your pons, and your medulla is right there. Has nothing to do with alligators being angry. It has everything to do with heart rate. So that's the only part. Not all organisms have this. Depending on what their senses are, some areas for like the frog brain, for example, have two big lobes on their brain. It's kind of interesting. Let me show you a picture of that. For example, frog brain. Pig brains, so here's the frog brain. I thought was kind of interesting. So this is an optic. These are the optic lobes here, and your olfactory lobes here, which would be smell, and here is sight. But look at how very simple this brain is, and it's tiny. Actually, it's very, very small. Thalamoencephalon. These brains are really, so you can kind of see the location. To be able to get a frog brain out, it is a real challenge. Let me look at this picture here. This is kind of cool. So here, as you can see, again, you'd have to cut through the skull to be able to see it, but look at how tiny, look at how ginormous those optic lobes are. So you think about, well, what does a frog really have to work on? Well, you've got huge optic lobes, and then you have your olfactory lobes. We don't have any optic lobes. The whole back of our head is an optic lobe, which I thought was kind of interesting. Let me see if I can do see if they'll show me like an insect. So here's an internal anatomy. Let me blow this one up. Internal anatomy of a grasshopper. Oh, sure. Now it's, oh, here we go. So even here's the optic lobes of the brain, and here's the brain working down. So very similar to the spinal cord of organisms that we look, but look at, here's the optic. So think about it. I mean, a grasshopper is really relying on sight to worry about surviving. Let's see if I can go back. Well, let me go back. Let's see, what other brain do we want to look at? Oh, how about a worm? Earthworm brain. Well, here's the inside of an earthworm. They can barely see it. Here is the cerebral ganglion. This is the like pharynx, but the cerebral ganglion is just, oh, there it is. There, it's tiny right there. I was trying to see if I could get a bigger, oh, right here. So even an earthworm has a brain. And think about, what would an earthworm, what is it going to really worry about? And the brain for that is really a sensory. It's a photoreceptor, because light means scary. I could be killed. So that's what it's mainly. And there's tons of different pictures of dissections here. So overall on the brain, make sure you look at the virtual brain, know the basics of the anatomy of the brain, just medulla and cerebrum and things like that. Think about how things can be different. And look over all of that synapse stuff. We did also go over 40.2 to 40.4, and I just went over some basic terms, and we talked about being warm-blooded versus cold-blooded in class. See you later.